Welcome to another episode from the Veterinary Research Channel. Today, we delve into fascinating research concerning a common health issue in one of our beloved canine companions, the Miniature Schnauzer. This study, published in the Journal of Veterinary Internal Medicine in 2024, explores the complexities of hypertriglyceridemia or high triglyceride levels in the blood within this particular breed. As many of you know, Miniature Schnauzers have a notable predisposition to this condition, which can unfortunately pave the way for serious health problems. These can include life-threatening pancreatitis, the formation of gallbladder mucoceles, and even glomerular proteinuria, a condition affecting the kidneys. Traditionally, it was thought that primary hypertriglyceridemia in miniature schnauzers, meaning high triglycerides without an identifiable underlying cause, was a single entity, primarily characterized by elevated triglyceride-rich lipoproteins and lower levels of low-density lipoproteins. However, something didn't quite add up. Veterinary professionals observed that miniature schnauzers diagnosed with presumed primary hypertriglyceridemia exhibited a wide range of responses to typical management strategies, such as dietary changes involving a low-fat content. Some dogs would see their triglyceride levels return to a normal range, while others showed little to no improvement. This variability hinted at a more intricate picture, suggesting that perhaps what we considered a single condition might actually encompass multiple underlying subtypes. This research team hypothesized that there could indeed be different forms or phenotypes of primary hypertriglyceridemia within the miniature Schnauzer breed. To investigate this, they embarked on a study to identify and characterize these potential subtypes by using a sophisticated technique called cluster analysis. This method allows researchers to group individuals based on similarities in their data, in this case, the detailed makeup of their lipoprotein profiles. The goal was to see if distinct patterns emerged that could differentiate dogs with hypertriglyceridemia. The study involved a careful selection of 50 miniature schnauzers. 20 of these dogs had normal serum triglyceride concentrations, serving as a control group. 25 were identified as having primary hypertriglyceridemia, meaning their high triglyceride levels weren't attributable to another underlying disease. Finally, a smaller group of 5 dogs had secondary hypertriglyceridemia, where the elevated triglycerides were linked to a known condition, such as diabetes mellitus or hypothyroidism. The researchers utilized archived serum samples from these dogs. To gain a comprehensive understanding of their lipid profiles, they employed a continuous lipoprotein density profiling method. This technique separates the various types of lipoproteins in the blood based on their density, providing a detailed snapshot beyond just the total triglyceride concentration. This method can identify 11 distinct density-based lipoprotein fractions in dogs. While these fractions are given nominal classifications such as triglyceride-rich lipoprotein and low-density lipoprotein, based on what types of lipoproteins are most likely to be found in those density ranges, the precise composition and functional roles of these subfractions in dogs are still being actively investigated. Once the lipoprotein profiles were generated, the researchers used a statistical approach called hierarchical cluster analysis to identify natural groupings within the data. They then compared clinical information, such as the dog's age, sex, body condition score, and the fat content of their diet, across these identified clusters to see if any of these factors could explain the groupings. Now let's move on to the exciting part: the results of this analysis. The cluster analysis successfully identified 6 distinct clusters of miniature schnauzers based on their lipoprotein profiles. Intriguingly, the dogs with primary hypertriglyceridemia were not confined to a single group but were distributed across 4 of these 6 clusters. One particular cluster stood out, showing the highest levels of intensity for both triglyceride-rich lipoprotein and low-density lipoprotein fractions. Interestingly, this cluster also included 4 of the 5 dogs with secondary hypertriglyceridemia. This suggests that these dogs, regardless of whether their high triglycerides were primary or secondary, shared a similar pattern of lipoprotein elevation. 2 other clusters were characterized by moderately high triglyceride-rich lipoprotein levels, but showed low to intermediate intensities of low-density lipoproteins. This indicates that while triglycerides were elevated, the low-density lipoprotein component was not as pronounced in these groups. The 4th cluster, containing dogs with primary hypertriglyceridemia, presented a different picture altogether. These dogs had high levels of low-density lipoprotein but showed variable levels of triglyceride-rich lipoprotein. What was also noteworthy about this cluster was that it contained an equal number of dogs with normal triglyceride concentrations and dogs with mild hypertriglyceridemia. This highlights that relying solely on a single triglyceride measurement might not capture the full spectrum of lipoprotein abnormalities. Finally, the remaining 2 clusters were composed exclusively of dogs with normal triglyceride concentrations. These dogs exhibited low levels of triglyceride-rich lipoproteins and low to intermediate levels of low-density lipoproteins. These clusters likely represent variations in the normal lipoprotein profiles within the Miniature Schnauzer breed. When the researchers compared the clinical data across these 6 clusters, they did not find any apparent links between the clusters and factors like age, sex, body condition score, or dietary fat content. This suggests that the identified lipoprotein profile subtypes are likely driven by other underlying mechanisms, possibly genetic factors or other physiological differences not measured in this study. The researchers also noted that while serum cholesterol concentrations were not a primary focus of the study, available data for some of the dogs indicated variability across the groups. Some dogs with hypertriglyceridemia also had elevated cholesterol, while others did not. Now, let's delve into the discussion of these findings and their implications. The fact that the study identified 6 distinct clusters of lipoprotein profiles in miniature schnauzers, and that dogs with primary hypertriglyceridemia were spread across 4 of them, strongly supports the initial hypothesis. There appears to be a spectrum of lipoprotein phenotypes within this breed that cannot be accurately predicted by simply measuring the concentration of triglycerides in the blood. The first major division observed in the lipoprotein profiles was primarily driven by differences in the low-density lipoprotein 4 and low-density lipoprotein 5 fractions. Dogs in one major grouping had higher levels of these 2 fractions compared to the other group, and both of these major groupings contained dogs with primary hypertriglyceridemia. This observation is important because in healthy dogs, both very low-density lipoproteins, which are part of the triglyceride-rich lipoprotein family, and low-density lipoproteins contribute significantly to overall triglyceride concentrations. This helps explain why a high triglyceride level alone doesn't tell us whether the elevation is primarily in triglyceride-rich lipoproteins, low-density lipoproteins, or a combination of both. The largest cluster identified in the study, cluster B2, was particularly interesting as it included a significant proportion of dogs with primary hypertriglyceridemia and a large majority of the dogs with secondary hypertriglyceridemia. The presence of both primary and secondary cases in the same cluster could suggest that the study, with its limited number of secondary hypertriglyceridemia cases, might not have had enough power to distinguish unique lipoprotein patterns associated with specific underlying conditions like hypothyroidism or diabetes mellitus. While it's generally understood that conditions like hypothyroidism and diabetes mellitus can lead to increases in all lipoprotein fractions, and hyperadrenocorticism often primarily increases low-density lipoprotein fractions, the methodologies used in this study to define the subfractions differ from those in previous research, making direct comparisons challenging. Notably, within this cluster, 3 of the secondary hypertriglyceridemia dogs, 1 with hypothyroidism and 2 with diabetes mellitus, showed very similar lipoprotein profiles, characterized by high triglyceride-rich lipoprotein and low-density lipoprotein levels but not high-density lipoprotein. Interestingly, 1 dog with diabetes mellitus-related secondary hypertriglyceridemia had a lipoprotein profile that didn't even fall within the major grouping that contained most other hypertriglyceridemia cases. This raises the possibility that in some instances, an underlying genetic predisposition to primary hypertriglyceridemia might be the main driver of high triglycerides, even if the dog also has a condition known to cause it. The fact that some of the dogs later diagnosed with endocrinopathies had already shown mild elevations in triglycerides suggests that multiple factors likely contribute to the development of hypertriglyceridemia. It's important to remember that high triglyceride levels are not a guaranteed outcome of conditions like diabetes mellitus, hypothyroidism, or hyperadrenocorticism. In humans, hypertriglyceridemia is viewed as a complex condition influenced by a combination of genetic and environmental factors. Another potential explanation for the clustering of primary and secondary cases is the possibility that some dogs categorized as having primary hypertriglyceridemia might have had an undiagnosed, subclinical endocrinopathy. While the researchers did perform thyroid and adrenal function tests on most of the primary hypertriglyceridemia dogs in this cluster, the fact that not all dogs underwent comprehensive endocrine screening is a limitation of the study. The study also identified 2 clusters, A1 and A4, that were predominantly composed of dogs with hypertriglyceridemia and showed a pattern of higher triglyceride-rich lipoproteins without significant elevations in low-density lipoproteins. Cluster A1 consisted of mild hypertriglyceridemia cases with low to intermediate triglyceride-rich lipoprotein and low-density lipoprotein levels. Cluster A4 showed low to intermediate low-density lipoprotein levels and included dogs with a range of hypertriglyceridemia severity. These patterns are somewhat consistent with what previous research had described in miniature schnauzers with normal and high triglyceride levels, where hypertriglyceridemic dogs often showed higher triglyceride-rich lipoproteins and lower low-density lipoproteins compared to other breeds. This study further reinforces that triglyceride concentration alone is not a perfect indicator of underlying lipoprotein metabolism, as some dogs with normal triglyceride levels were found within the main hypertriglyceridemia clusters. The differences observed between cluster B2, which showed elevations in both triglyceride-rich lipoproteins and low-density lipoproteins, and clusters A1 and A4 with primarily elevated triglyceride-rich lipoproteins, suggest that different mechanisms might be at play in the development of hypertriglyceridemia in these groups. These mechanisms could be genetic, environmental, or a combination of both. For instance, deficiencies in enzymes like lipoprotein lipase or hepatic lipase are known to be associated with distinct patterns of lipid and lipoprotein abnormalities. While the clinical variables evaluated in the study did not differ significantly between the clusters, important factors such as insulin resistance, central obesity, a history of pancreatitis, and exercise levels were not assessed and could contribute to these differences. Insulin resistance is of particular interest given its known association with hypertriglyceridemia in miniature schnauzers. Pancreatitis, also linked to hypertriglyceridemia in this breed, is characterized by specific changes in lipoprotein profiles that differ from those identified as the primary drivers of the clusters in this study. Furthermore, different types of dietary fats which can influence lipoprotein metabolism were not specifically evaluated. Interestingly, the study also identified two clusters, A2 and A3, that comprised only dogs with normal triglyceride levels. Both of these clusters had low triglyceride-rich lipoprotein levels but differed in their low-density lipoprotein intensities, with cluster A2 generally showing lower levels. Lower low-density lipoprotein levels have been previously noted as a characteristic of the miniature Schnauzer breed. As this study did not include other breeds for comparison, it's difficult to definitively say whether the patterns observed in clusters A2 and A3 represent normal variations within healthy miniature Schnauzers, or if cluster A2 might indicate a breed-specific lipid profile even in dogs with normal triglyceride concentrations. The remaining cluster, B1, contained a roughly equal mix of dogs with normal and mild hypertriglyceridemia. These dogs showed variable triglyceride-rich lipoprotein levels and higher levels of low-density lipoprotein 4 and 5, and less consistently, low-density lipoprotein 1 through 3. One possibility is that the dogs in cluster B1 represent an early or milder form of the dyslipidemia phenotype observed in cluster B2. A limitation of the study is that cholesterol concentrations were not measured in all dogs, particularly in the normal triglyceride group, so it's possible that some dogs in this or other clusters had hypercholesterolemia despite normal triglyceride levels. It's also conceivable that the mild hypertriglyceridemia in the dogs in cluster B1 was due to other unmeasured dietary or patient factors rather than genetic predisposition. The study found no significant differences in dietary fat content across the clusters. While feeding a low-fat diet can resolve hypertriglyceridemia in some miniature Schnauzers and alter their lipoprotein profiles, the 10 dogs in this study that were on low-fat diets appeared to cluster with other dogs having similar triglyceride concentrations. Similarly, age, sex, and body condition score did not appear to be significant differentiating factors between the clusters, possibly due to the study's sample size and the relatively narrow range of these variables within the study population. The researchers acknowledged that the study's relatively small sample size, especially for dogs with moderate to severe hypertriglyceridemia, is a limitation. A larger study could potentially reveal even more distinct subtypes and provide a more comprehensive understanding of hypertriglyceridemia in this breed. Furthermore, the current limited understanding of the precise composition and function of the density-based lipoprotein subfractions in dogs restricts the ability to fully interpret the driving factors behind the observed differences. Future research correlating genetic variations with these specific lipoprotein profile clusters could potentially uncover the genetic underpinnings of these hypertriglyceridemia subtypes, including a larger number of dogs with secondary hypertriglyceridemia both within the miniature Schnauzer breed and in other breeds not prone to primary hypertriglyceridemia, could help to better distinguish the effects of underlying endocrinopathies from those caused by genetic risk factors or a combination of both. Finally, it's important to consider that the lipoprotein profiles obtained in this study represent a single point in time, and triglyceride concentrations can fluctuate considerably in miniature Schnauzers with hypertriglyceridemia even under consistent conditions. In conclusion, this study provides compelling evidence for the existence of multiple lipoprotein profile patterns in miniature Schnauzers, suggesting that hypertriglyceridemia in this breed is not a single entity but rather a spectrum of conditions. The findings indicate that differences in low-density lipoprotein fractions might be a key factor in distinguishing these potential subtypes. Further research is clearly warranted to confirm the range and number of these distinct lipoprotein profiles and to investigate their origins and potential clinical consequences. Lipoprotein profiling holds promise as a valuable tool for future studies aimed at unraveling the complexities of hypertriglyceridemia in miniature schnauzers and potentially leading to more targeted diagnostic and therapeutic approaches. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.